Hi class, this is your instructor Scholar Huff, and we're now in the 32nd chapter, Animal Diversity and the Evolution of Body Plans. Animals are among the most abundant living organisms, found in almost every habitat. They bewilder us with their diversity of form, habitat, behavior, and lifestyle. About 1.5 million species have been described, and several million more are thought to await discovery. Despite their great diversity, animals have much in common. For example, locomotion is a distinctive characteristic, although not all animals can move about. Early naturalists thought that sponges and corals, like the yellow tube sponge shown above, were plants, because the adults are attached to the surface on which they live. In this chapter, and chapters 33 and 34, we explore the great diversity of modern animals, the result of long evolutionary history. We start in this chapter with a discussion of the features that characterize animals and review the phyla at the base of the animal phylogeny. In chapters 33 and 34, we discuss the two major groups of animals, the protostomes and deuterostomes. Let us begin. Animals are so diverse that few criteria fit them all. But some, such as animals being eaters, or should I say consumers, apply to all. Others, such as animals being mobile, they can move about, have exceptions. Taken together, the universal characteristics and other features of major importance exhibited by most species are convincing evidence that animals are a monophyletic group. All members more closely related to one another than any members are to another type of organism. This conclusion has been confirmed when DNA data have been used to build phylogenies of all living organisms. Animals share some general characteristics. Heterotrophy. All animals are heterotrophs, that is, they obtain energy and organic molecules by ingesting other organisms, unlike autotrophic plants and algae. Animals cannot construct organic molecules from inorganic chemicals. Some animals, herbivores, consume autotrophs. Other animals, carnivores, consume heterotrophs. Some animals, omnivores, consume both autotrophs and heterotrophs, and still others, detritivores, consume decomposing organisms. Multicellularity. All animals are multicellular. Many have complex bodies. Unicellular heterotrophic animals, which were at one time regarded as simple animals, are now considered members of several different clades within the large and diverse group of protists. No cell walls. Animal cells differ from those of other multicellular organisms. They lack rigid cell walls and are usually quite flexible. The many cells of animal bodies are held together by extracellular frames of structural proteins such as collagen. Other proteins form unique intercellular junctions between animal cells. Active movement. Although other single-celled organisms are able to travel from place to place, many animals move more rapidly. And note I said many animals move rapidly and in more complex ways. This ability is perhaps their most striking characteristic, one directly related to the flexibility of their cells and the evolution of muscle tissues and nerve tissue. A remarkable form of movement unique to animals is flying, a capability that is well developed among vertebrates and insects, such as the butterfly, shown in figure 32.1a. Although many animals cannot move from place to place, they are sessile, or do so rarely or slowly. They are sedentary. They have muscles or muscle fibers that allow parts of their bodies to move. But note, sponges, however, have little capacity for movement. Diversity of form. Animals vary greatly in form, exhibiting a diversity of body plans, ranging in size from organisms too small to see with the unaided eye to enormous whales and giant squids. Almost all animals lack a backbone. They are therefore called invertebrates, like the millipede shown in figure 32.1b. Of the 1.5 million known living animal species, fewer than 60,000 of those are vertebrates, those species that possess a backbone. Most of the many millions of animal species awaiting discovery are invertebrates. Diversity of habitat. Animals are grouped into 35 to 40 phyla, and no, I will not teach you all of those, most of which have members that occur only in the sea. Members of fewer phyla occur in freshwater, and members of fewer still occur on land. Members of 3 phyla that are successful in the marine environment, Arthropoda, Nematoda, and Chordata, also dominate animal life on land. Only one phylum, Onychophora, is entirely terrestrial. And I might mention the onychophores a little later in this same lecture. Sexual reproduction. Most animals reproduce sexually. Animal eggs, which are not mobile, are much larger than the small, usually flagellated sperm. In animals, cells formed in meiosis function as gametes. These haploid cells do not divide by mitosis first, as they do in plants and fungi, but rather fuse directly with each other to form the zygote. Consequently, there is no counterpart among animals to the alternation of haploid gametophyte and diploid sporophyte generations, characteristic of plants. Some individuals of some species and all individuals of a very few others are incapable of sexual reproduction. Embryonic development. Shortly after fertilization, an animal zygote first undergoes a series of mitotic divisions called cleavage. And as in the dividing frog's eggs in the figure 32.1c, cleavage produces a ball of cells, the blastula. In most animals, the blastula folds inward at one point to form a hollow sac with an opening at one end called the blastopore. An embryo at this stage is called a gastrula, going through gastrulation. The subsequent growth and movement of the cells of the gastrula differ from one group of animals to another, reflecting the evolutionary history of the group. Embryos of most kinds of animals develop into a larva which looks unlike the adult of the species, lives in a different habitat, and eats different sorts of food. In most groups, it is very small. A larva undergoes metamorphosis, a radical reorganization to transform into the adult body form. Lastly, it says tissues, though I will say cells instead of tissues. The cells of all animals except sponges. The cells of all animals except sponges are organized into structural and functional units called tissues, collections of cells that together are specialized to perform specific tasks. So yes, it is the cell that comprises the body that is specialized to perform specific functions. These are the characteristics of all animals. And here are the pictures referred to a bit earlier with the butterfly, and then thereafter the millipede, and then ultimately the blastula. The features described just a moment ago evolved over the course of millions of years. We can understand how the evolution of life has proceeded by examining the types of animal bodies and body plans present in fossils and in existence today. 5 key innovations can be noted in animal evolution. 1 is symmetry. Thereafter, tissues, the body cavity, various patterns of embryonic development, and then segmentation or repeated body units. These innovations are explained in this section, so let's get to them. A typical sponge lacks definite symmetry. They are asymmetrical, growing as an irregular mass. Virtually all other animals have a definite shape and symmetry that can be defined along an imaginary axis drawn through the animal's body. The two main types of symmetry are radial and bilateral. And note that many animals are not perfectly symmetrical, but close enough that they are considered symmetrical. Radial symmetry is exhibited by members of the phylum Cnidaria, composed of jellyfish, sea anemones, and corals. Cnidarian parts are arranged in such a way that any longitudinal plane passing through the central axis divides the organism into halves that are approximate mirror images. A pie, for example, is radially symmetrical. You can cut it with a knife from above, and as long as you cut through the center, the two halves will be identical. The same is true for a sea anemone. I'll show you this in a moment. Well, I'll show it now. So here at the top, figure 32.2a, radial symmetry. And that is of a sea anemone, as I just mentioned. The bodies of most animals other than sponges and cnidarians exhibit bilateral symmetry, in which the body has right and left halves that are mirror images of one another. The clade-containing animals with this body plan is termed bilateria. The sagittal plane defines these halves, which you see now. The sagittal plane is there in kind of a teal color. In addition to the left and right halves, a bilaterally symmetrical body has dorsal and ventral portions, which are divided by the frontal plane, and the anterior and posterior ends, which are divided by the transverse plane. So you can see the frontal plane there kind of in yellow, and then you have that transverse plane kind of in that tan color. Sometimes deciding whether an organism should be considered bilaterally or radially symmetrical is not easy. For example, echinoderms, which include the sea stars and their relatives, adults are radially symmetrical, and I'll actually call this pentaradial symmetry later on because the body has 5 clear sections, but the larvae are bilaterally symmetrical, and I'll make sure to emphasize this a bit later on. Bilateral symmetry constitutes a major evolutionary advance in the animal body plan. Bilaterally symmetrical animals have the ability to move through the environment in a consistent direction, a feat that is difficult for radially symmetrical animals. Associated with this directional movement is the grouping of nerve cells into a brain and location of sensory structures such as eyes and ears at the anterior end of the body. This concentration of nervous tissue at the anterior end, which appears to have occurred early in evolution, is called cephalization. Much of the layout of the nervous system in bilaterally symmetrical animals is centered in one or more major longitudinal nerve cords that transmit information from the anterior sense organs and brain to the rest of the body. Cephalization is often considered a consequence of the development of bilateral symmetry. The zygote, which is a fertilized egg, has the capability of giving rise to all the kinds of cells in an animal's body. That is, it is totipotent. Totipotent means literally all-powerful. During embryonic development, cells specialize in carrying out particular functions. In all animals except sponges, the process is irreversible. Once a cell differentiates to serve a function, it and its descendants can never serve any other. A sponge cell that had specialized to serve one function such as lining the cavity where feeding occurs, can lose the special attributes that serve that function and change and serve another function, such as being a gamete. Thus, a sponge cell can de-differentiate and redifferentiate, but this ability comes at a cost. Cells of all other animals are organized into tissues, each of which is characterized by cells of particular morphology and capability. But the ability of sponge cells to differentiate prevents them from becoming specialized to clearly form defined tissues. Sponges are unique in being able to de-differentiate and then re-differentiate. The fact that all other animals differentiate irreversibly suggests that animals with bodies containing cells specialized to serve particular functions may have an advantage over those with cells that potentially have multiple functions. The evolution of specialized tissues may be a key innovation, a trait that fosters evolutionary diversification. In the process of embryonic development, the cells of most types of animals organize into 3 layers called germ layers: an outer ectoderm, an inner endoderm, and an intermediate or middle mesoderm. Animals with all 3 embryonic germ layers are said to be triploblastic. During maturation from the embryo, certain organs and organ systems develop from each germ layer. The ectoderm gives rise to the outer covering of the body and the nervous system. The endoderm gives rise to the digestive system, including the intestines. The skeleton and muscles develop from the mesoderm. All triploblastic animals are members of Bilateria. Cnidarians are diploblastic and have only 2 germ layers, the endoderm and ectoderm, and lack organs. Sponges lack germ layers altogether. They, of course, have no tissues or organs. A key innovation in the body plan for some bilaterians is the body cavity, isolated from the exterior of the animal. This is different from the digestive cavity, which is open to the exterior at least through the mouth, and in most animals, at the opposite end as well via the anus. The evolution of efficient organ systems within the animal body was not possible until a body cavity evolved for accommodating and supporting organs, such as the heart and lungs, distributing materials and fostering complex developmental interactions. The cavity is filled with fluid in most animals. The fluid is liquid, but in vertebrates it is gas. A few types of bilaterians have no body cavity, the space between the tissues that develop from the mesoderm and those that develop from the endoderm being filled with cells and connective tissue. These are so-called acoelomate animals. Excuse me. Body cavities appear to have evolved multiple times in Bilateria. In some animals, a body cavity is called the pseudocoelom, develops embryologically between the mesoderm and the endoderm, and thus in the adult between tissues derived from the mesoderm and those derived from the endoderm. Animals with this type of body cavity are called pseudocoelomates. Although the word pseudocoelom means false coelom, this is a true body space and characterizes many groups of animals. A coelom is a cavity that develops entirely within the mesoderm. The coelom is surrounded by a layer of epithelial cells derived from the mesoderm, is termed peritoneum. So here you see the acoelomates. So they have no body cavity. Pseudocoelomates have a body cavity. It's just not completely lined. And finally, coelomates. They have a completely lined body cavity. In many small animals, nutrients and oxygen are distributed and wastes are removed by fluid in the body cavity. Most large animals, in contrast, have a circulatory system, a network of vessels that carry fluids to and from the body parts distant from the sites of digestion and gas exchange. The circulating fluid carries nutrients and oxygen to the tissues and removes wastes, including carbon dioxide, by diffusion between the circulatory fluid and the other cells of the body. In an open circulatory system, the blood passes from vessels into sinuses, which are open spaces within the body, and mixes with body fluid that bathes the cells of tissues, then re-enters vessels in another location. In a closed circulatory system, the blood flows entirely within blood vessels, so it is physically separated from other bodily fluids. Blood moves through a closed circulatory system faster and more efficiently than it does through an open system. Open systems are typical of animals that are relatively inactive and so do not have a high demand for oxygen. In small animals, blood can be pushed through a closed circulatory system by the animal's movement. In larger animals, the body musculature does not provide enough force, so the blood must be propelled by contraction of one or more hearts, which are specialized muscular parts of the blood vessel. The process of embryonic development in animals are discussed fully in the chapter called 52. So briefly, development of bilaterally symmetrical animal begins with mitotic cell divisions called cleavage. And those mitotic cell divisions of the egg called cleavage that lead to the formation of a hollow ball of cells, which subsequently indents to form a two-layered ball. The internal space that is created through such indentation is the archenteron. It communicates with the outside by a blastopore. In a protostome, the mouth of the adult animal develops from the blastopore or from an opening near the blastopore. And protostome means first mouth. The first opening becomes the mouth. Protostomes includes most bilaterians, including the flatworms, the nematodes, the mollusks, annelids, and arthropods. In some protostomes, both mouth and anus form from the embryonic blastopore. In other protostomes, the anus forms later in another region of the embryo. Two outwardly dissimilar groups, the echinoderms and the chordates, together with a few other small phyla, constitute deuterostomes. And the deuterostomes are those in which the mouth of the adult animal does not develop from the blastopore. The deuterostome blastopore gives rise to the organism's anus, and the mouth develops from a second pore that arises later in the development. Deuterostome means second mouth. Protostomes and deuterostomes differ in several other aspects of embryology too. The cleavage pattern relative to the embryo's polar axis, which runs from top to bottom through the center of the organism, determines how the resulting cells will lie with respect to one another. Two patterns of cleavage characterize most major clades of animals. In one clade of protostomes, the Lophotrochozoa, each new cell cleaves off to the right or the left, producing a layer of cells shifted to one side or the other, relative to those from which they cleaved. This pattern is called spiral cleavage because a line drawn through a sequence of dividing spirals of cells spirals outward from the polar axis. Spiral cleavage is characteristic of annelids, mollusks, nematarians, and related phyla. And that's nemerteins, By contrast, in all deuterostomes and convergently in a few protostomes, such as the brachiopods, the cells divide parallel and at right angles to the polar axis. As a result, the pairs of cells from each division are positioned directly above and below one another, a process that gives rise to a loosely packed ball. This pattern is called radial cleavage because a line drawn through a sequence of dividing cells describes a radius outward from the polar axis. Many protostomes exhibit determinate development, in which the type of tissue each embryonic cell will form in the adult is determined early. In many lineages, even before cleavage begins, when the molecules that act as developmental signals are localized in different regions of the egg. Consequently, each embryonic cell is destined to occur only in particular parts of the adult body. So, if the cells are separated or lost, development cannot proceed. Deuterostomes, conversely, display indeterminate development. The first cell divisions of the zygote produce identical daughter cells. If the cells are separated, anyone can develop into a complete organism. This is how identical twins are formed. This is also how, by way of in vitro fertilization, you can remove a cell from an embryo for further analysis. In other words, you would freeze an embryo, and then you can run a battery of tests by way of a PGD, that pre-genetic implantation diagnosis, using fluorescent in situ hybridization to determine of the embryos which embryo is, I won't just say made whole, but I would say has no genetic anomalies or no variants. Thus, each cell remains totipotent and its fate is not determined for several cleavages. The coelom arises within the mesoderm, and protostomes cells simply move apart from one another to create an expanding coelomic cavity within the mass of mesodermal cells. In deuterostomes, groups of cells pouch off the end of the archenteron, which you may recall is the primitive gut, the hollow center of the developing embryo that is lined with endoderm. And this shows you everything I've given you. So at the bottom in green, you have the deuterostomes. At the top, you have the Lophotrichozoan protostomes in the gray or blue. Looks like gray. Segmented animals consist of a series of linearly arrayed compartments that typically look alike, at least early in development, but that may have specialized functions. Development of segmentation is mediated at the molecular level by Hox genes. During early development, segments first are obvious in the mesoderm, but later are reflected in the ectoderm and endoderm. Two advantages result from early embryonic segmentation. In highly segmented animals, such as the earthworms from phylum Annelida, each segment may develop a more or less complete set of adult organ systems. Because these are redundant systems, damage to any one segment need not be fatal because other segments duplicate the damaged segment's functions. And then locomotion is more efficient when individual segments can move semi-independently. Because partitions isolate the segments, each can contract or expand independently. Therefore, a long body can move in ways that are often quite complex. Segmentation underlies the origination of body plans of the most morphologically complex animals. In some adult arthropods, the segments are fused, but the segmentation is usually apparent in embryological development. In vertebrates, the backbone and muscle blocks are segmented, although segmentation is often disguised in the adult form. Previously, zoologists considered that true segmentation was only found in annelids, arthropods, and chordates, but segmentation is now recognized to be more widespread. Traditionally, the phylogeny of animals has been inferred using features of anatomy and aspects of embryological development, from which a broad consensus emerged over the last century concerning the main branches of the animal tree of life. However, in the past 30 years, gene sequence data have accumulated at an accelerating pace for all animal groups. Phylogenies developed from different molecules sometimes suggest quite different evolutionary relationships among the same groups of animals. However, combining data from multiple genes has resolved the relationships of most phyla. Current studies are using sequences from hundreds of genes and even entire genomes to try to fully resolve the animal tree of life. The diversity of animals is obvious in tables 32.1 and 32.2, which describe key characteristics of the 21 animal phyla. Although they differ from one another in some respects, phylogenies utilizing molecular data share some deep structure with the traditional animal tree of life. Figure 32.5 is a summary of the animal phylogeny developed from morphological, molecular, life history, and other types of relevant data. Some parts of this phylogeny are not firmly established, and new studies are constantly appearing, often with somewhat different conclusions. It is an exciting time to be a systemicist, but shifts in understanding of relationships among groups of animals can be frustrating to some. Like any scientific idea, a phylogeny is a hypothesis open to challenge and to being revised in light of additional data. So before I go to classification of animals, I want to stop here for table 32.1. And this goes through the flatworms in phylum Platyhelminthes. I recommend as you study, you at least look at A, the phylum, B, typical examples, C, what the typical animal looks like, D, key characteristics. As far as tests are concerned, I will not ask about an approximate number of species, though there's a caveat. You should know that the arthropods in phylum Arthropoda have over 1 million species that have been named and classified. So next is Mollusca, Chordata, and Platyhelminthes. And yes, that is Platyhelminthes, the flatworms. And at Chordata, this list here, there are invertebrate chordates which are not listed there. I think about the tunicate as being one such example. And now we have phylum Nematoda, phylum Annelida, and phylum Porifera. Please take your time to go through these first 4 columns. So here is the other side. So now we have the echinoderms, cnidarians from phylum Cnidaria, and echinoderms are from phylum Echinodermata, and then phylum Bryozoa. And now we have phylum Rotifera, Nematia, Tardigrada, and Brachiopoda. And we have Ctenophora, Catognatha, Onychophora, and then Loracifera. And note, these numbers are getting smaller and smaller and smaller. So we have Cycliophora, Placozoa, and Micronathozoa. And note, the Micronathozoa were discovered roughly 26 years ago. So note, animals are still being discovered. So one consistent result is that Porifera, the sponges, constitutes a monophyletic group. Some systemicists had considered sponges to comprise 2 or 3 groups that are not particularly closely related, but molecular data support what had been the majority view. That phylum is monophyletic. Among remaining animals, termed Eumetazoa molecular data are in accord with the traditional view that cnidarians, which includes the hydras, the sea jellies, and corals, branch off the tree before the origin of animals with bilateral symmetry, the bilateria. Our understanding of the phylogeny of the deuterostome branch of bilateria has not changed much in recent times, but our understanding of the phylogeny of protostomes has been altered by molecular data. The most revolutionary development is that annelids and arthropods, which have been considered close related because they both exhibit segmentation, are in fact not close relatives, but rather belong to separate clades. Now arthropods are grouped with the protostomes that molt their cuticles at least once during their life. These are termed excidiozoans, which means molting animals. Molecular sequence data can help test our ideas with morphological features, reveal evolutionary relationships best. In this case, molecular data allowed us to see the contrary to our hypothesis. Segmentation seems to have evolved convergently, but molting did not. So annelids and arthropods are now there. So previous phylogenies were based on the evolution of the coelom. It is now clear that the state of the internal cavity has evolved many times. The coelom appears to have evolved once. Some coelomate clades have reverted to acoelomate or pseudocoelomate condition, and all deuterostomes have body cavities. And the protostomes, they are variable, as we just discussed. Early in their history, protozoans divided into two clades, the Lophotrochozoans and the Excidiozoans. The Lophotrochozoans grow by gradual addition of mass to the body and undergo spiral cleavage. The Excidiozoans are animals that molt, a phenomenon that seems to have evolved only once in the animal kingdom. Of the numerous phyla of protostomes assigned to the Excidiozoa, Arthropoda contains the largest number of described species of any phylum. Deuterostomes consist of fewer phyla and species than protostomes, and are more uniform in many ways, despite great differences in appearance. Echinoderms, such as sea stars, and chordates, such as humans, share a mode of development that is evidence of their evolution from a common ancestor and separates them clearly from other animals. So this is that proposed revision to the animal tree of life that I've been discussing. So a phylogeny of many of the 35 to 40 phyla reflects a consensus based on the interpretation of anatomical and developmental data, as well as results derived from molecular phylogenetic studies. So here you have them. These are the animals. So the modern phylogeny. And then we have our protist ancestor, the coenoflagellates, giving rise to the animals by way of metazoa. So we have the porifera, the ctenophora. Now we have placozoa, cnidarians, aceola, the micronazoa, rotifera, the chaetonactiths, the platyhelminths, cycliophora, mollusca, annelida, bryozoa, brachiopoda, nemertea, loricifera, and then we have nematoda. And then finally, we have the Nicornica, the arthropoda, onychophora, tardigrada, echinodermata, and chordata. These are the animals. As this chapter prepares to get a bit more specific in animals, let us begin. So we'll explore the great diversity of animals. We start with morphologically simplest members of the animal kingdom: sponges, jellyfish, and some types of worms. Fully a third animal phylum are discussed on a wormy body plan. So despite their simplicity, these animals can carry out all the essential functions of life, just as do morphologically complex animals: eating, respiring, reproducing, and protecting themselves. The major organization of the animal body first evolved in these animals, the basic body plan from which all the rest of animals evolve. Systemicists traditionally divide the animal kingdom Animalia, into two main branches, and kingdom Animalia is also referred to as the metazoa, as I earlier mentioned. So parazoa comprises animals that, for the most part, lack definite symmetry and do not possess tissues. These are sponges, phylum Porifera. Because they are so different in so many ways from other animals, some scientists inferred that sponges were not closely related to other animals, which would mean that what we consider animals had two separate origins. Eumetazoa are animals that have a definite shape and symmetry. All have tissues, and most have organs and organ systems. Now, most systemicists agree that parazoa and eumetazoa are descended from a common ancestor, so animal life had a single origin. Yes, animals are parsimonious. Phylogenies constructed with molecular data are divided about the positions of Porifera and Ctenophora. Many recent studies place Ctenophora as the sister taxon to all other animals, indicating that Porifera occurs within eumetazoa rather than it being its sister taxon. If this phylogenetic arrangement is correct, then specialized tissues may have evolved independently in ctenophores and other animals. Alternatively, the lack of specialized tissues in sponges may be the result of evolutionary loss of an ancestor with specialized tissues. Despite their morphological simplicity, like all animals, sponges, the phylum Porifera, are truly multicellular. There are more than 26,000 species of fungus that live in the sea, and perhaps 150 species live in fresh water. Marine sponges occur at all depths and may be among the most abundant animals in the deepest part of the oceans. Although some sponges are small, no more than a few millimeters across, others may reach 2 meters or more in diameter. Most members of this phylum lack symmetry. Some have a low encrusting form and grow covering various sorts of surfaces. Others are erect, lobed, and some even complex patterns. As is true of many marine invertebrate animals, larval sponges are free swimming. After a sponge larva attaches to an appropriate surface, it metamorphoses into an adult and remains attached to that surface for the rest of its life. Thus, adult sponges are sessile, that is, they are anchored immobile on rocks or other submerged objects. Sponges define themselves by producing chemicals that repel potential predators and organisms that might overgrow them. As a result, pharmaceutical companies are interested in possibly using these chemicals for human applications. Lacking a head, appendages, a mouth, an anus, and organized internal structure, characteristic of all animals, a sponge at first seems to be little more than a mass of cells embedded in a gelatinous matrix. In fact, a sponge contains several cell types, each with specialized functions. This distinguishes sponges as truly multicellular, by contrast with colonial protists, which may form aggregates of cells, but all of whose cells are functionally identical, except for those reproductive cells. A unique feature of sponge cells is their ability to differentiate from one type to another, and to differentiate from a specialized state into an unspecialized one. If a sponge is put through a fine sieve or a coarse cloth, so that the cells are separated, they will seek each other out and reassemble the entire sponge, a phenomenon that does not occur in any other animal. A small anatomically simple sponge is vase-like in shape. I suppose I'll show that again. The walls of the vase have 3 functional layers. Facing the internal cavity are flagellated cells called choanocytes, or collar cells. A larger and more complex sponge has many small chambers connected by channels rather than by a single chamber. Once water has passed through the flagellated chamber, it travels through channels that converge at a large opening called an osculum, through which water is expelled from the sponge. The body of a sponge is bounded by an outer epithelium consisting of flattened cells, somewhat like those that make up the outer layers of animals and other phyla. Pores on the sponge allow water to enter the channels that course through its body, leading to and from flagellated chambers. The name of the phylum Porifera refers to these pores, which are also called ostia. A large sponge has multiple oscula, but they are far, far fewer than the number of ostia. Some epithelial cells are specialized to surround the ostia. They can contract when touched or exposed to appropriate stimuli, causing the ostia to close, thereby protecting the delicate inner cells from the entry of potentially harmful substances such as sand and noxious chemicals. Cells surrounding the individual ostia operate independently of one another. Because a sponge has no nervous system, actions cannot be coordinated across large distances. Between the outer and inner layers of sponges cells, the sponge consists mainly of a gelatinous, protein-rich matrix called the mesohyl, which contains various types of amoeboid cells and eggs. In many kinds of sponges, some of these cells secrete needles of calcium carbonate or silica known as spicules, or fibers of a tough protein called spongin. Spicules and spongin form the skeleton of the sponge, strengthening its body. The siliceous spicules of some deep-sea sponges can reach a meter in length. A genuine bath sponge is the spongin skeleton of a marine sponge. Artificial sponges made of cellulose or plastic are modeled on the body design of this animal, with the porous body adapted to contain large amounts of water. Each choanocyte resembles a protist with a single flagellum. The pressure created by the beating flagella in the cavity contributes to the circulating water that brings in food and oxygen and carries out waste. In large sponges, the inner wall of the body interior is convoluted, increasing the surface area and therefore the number of flagella. In such a sponge, 1 centimeter cubed of a sponge can propel more than 20 liters of water per day. Choanocytes also capture food particles from the passing water, engulfing and digesting them. Obviously, this arrangement restricts a sponge from feeding on particles considerably smaller than choanocytes, largely bacteria. Some sponges can reproduce asexually, simply by breaking into fragments, and each fragment is able to continue growing as a new individual. Sponge sperm are created by transformation of choanocytes, which are then released into the water, where they may be carried on to another sponge of the same species. When a sperm is captured by a choanocyte, it is carried to an egg cell, which is in the mesohyl. And in many sponges, development of the externally ciliated larva occurs within the mother. In sponges of other species, the fertilized egg is released into the water, where development occurs. Whether a fertilized egg or ciliated larva is released after a short platonic stage or drifting stage, the larva settles on a suitable substrate where it forms into an adult. Eumetazoa. These are the animals with true tissues. The Eumetazoa contain animals that evolved the first key transition in the animal body plan, distinct tissues. The embryonic cell layers differentiate into tissues of the body, giving rise to the body plan characteristic of each group of animals. Eumetazoa comprises 3 groups. The first two, Ctenophora and Cnidaria, are traditionally considered diploblasts and possess these 2 layers. The third group, Bilateria, are triploblasts. In bilaterians, the embryonic mesoderm, which lies between the ectoderm and endoderm, forms the muscles. Eumetazoans also evolved body symmetry, the two main types of which are radial and bilateral symmetry. Pelagic members of the small marine phylum Ctenophora range from spherical to ribbon-like and are known as comb jellies, sea walnuts, or sea gooseberries. Yes, I said gooseberries. Abundant in the open ocean, most of these animals are transparent and only a few centimeters long, but rare ones develop deeply pigmented and reach 1 meter in length. They propel themselves through the water with 8 rows of comb-like plates of fused cilia that beat in coordinated fashion, scattering the light so they appear like rainbows. Ctenophores are the largest animals that use cilia for locomotion. Many ctenophores are bioluminescent, giving off bright flashes of light that are particularly evident in the deep sea or on the surface of the ocean at night. Most ctenophores have 2 long retractable tentacles used to capture prey. The epithelium of the tentacles contains colloblasts, a type of cell that bursts to discharge a strong adhesive material on contact with animal prey. The bodies of these fascinating and simply constructed diploblastic animals are radially symmetrical, and these, I say, referring to the cnidarians. So they are radially symmetrical and made of distinct tissues, although they do not have organs. Despite having no reproductive, circulatory, digestive, or excretory systems, cnidarians reproduce, exchange gas, capture and digest prey, and distribute the resulting organic molecules to all their cells. A cnidarian has no concentration of nervous system that could be considered a brain or even a ganglion, which is a small group of nerve cells. Rather, its nervous system is a latticework of cells, having junctions like those of bilaterians. All cnidarians have nervous receptors sensitive to touch, and some have gravity and light receptors, which include a few species' image-forming eyes. Cnidarians capture their prey with nematocysts, microscopic intracellular structures unique to the phylum. Food is captured by a cnidarian and brought to the mouth by tentacles that ring the mouth. Most of the 10,000 species of cnidarians are marine. A very few live in fresh water. A cnidarian exhibits one of 2 body forms, the polyp and the medusa. A polyp is cylindrical with a mouth surrounded by tentacles at the end of the cylinder opposite of where it's attached, so the mouth part faces upward. Attachment in most solitary polyps is to a firm substrate, but polyps that are part of a colony attach to the mass of a common colonial tissue. A medusa is discoidal or umbrella-shaped, with the mouth part surrounded by tentacles on one side. Most are free-living in water, and typically the mouth part faces downward. These 2 seemingly quite different body forms share the same morphology. The mouth opens to a sac-like gastrovascular cavity and is surrounded by tentacles. The cnidarian body plan has a single opening leading to the gastrovascular space, which is the site of digestion. Most gas exchange and waste discharge, and in many cnidarians, formation of gametes. The body wall is composed of 2 layers, the epidermis, which covers the surfaces in contact with the outside environment, and the gastrodermis, which covers the gastrovascular cavity. Between these two layers is the mesoglia, which varies from acellular to being no more than glue holding gastrodermis to the epidermis. In Hydra, to thick, rubbery, with many cells in large medusae called jellyfish. The gastrovascular space also serves as a hydrostatic skeleton. A hydrostatic skeleton serves two of the roles that a skeleton made of bone does. It provides a rigid structure against which muscles can operate and gives the animal shape. For muscles to operate against it, the fluid-filled space must be closed sufficiently tight so the fluid is under pressure and does not escape when the muscles contract around the space. In addition to the hydrostatic skeleton of all polyps, those of many species build an exoskeleton of chitin or calcium carbonate around themselves in many colonial species. The skeleton links members of the colony, forming what we call coral. The polyps of smaller number of species secrete internal skeleton elements. Polyps of some groups of cnidarians, such as sea anemones, do not have no skeleton at all. All medusae are solitary and form no skeleton. Cnidarians exhibit considerable variation in life history. Some cnidarians occur only as polyps and others exist only as medusae, but alternate between these two phases. In general, in species having both polyp and medusa life cycle, the medusa forms the gametes. The sexes are separate, and individual is either male or female. An egg and sperm unite to form a zygote, which develops into a planktonic ciliated planula larva that metamorphoses into a polyp. The polyp produces the medusae asexually. Dispersal occurs in both medusae and larvae. In most species with such a life cycle, a polyp may also produce other polyps asexually. If they remain attached to one another, the result is a group referred to as a colony. Cnidarians capture food by the aid of pneumatocysts, microscopic stinging capsules unique to the phylum. Although often referred to as stinging cells, they are capsules, each secreted within a cell of a pneumatocyte. Cnidarians may be morphologically simple, but pneumatocysts are the most complex structures secreted by a single animal's cells. When appropriately stimulated, the closure at the end of the capsule springs open and the tube is emitted. Pneumatocyst discharge is one of the fastest cellular processes in nature. The mechanism of discharge of pneumatocysts is unknown, and several hypotheses for it have been proposed. Although the action of the pneumatocyst is often described as harpoon-like, the tubule actually everts or turns inside out. It may penetrate or wrap around an object. The tubules of some nematocysts are barbed, and some carry venom. In a very few species, the venom is strong enough to kill a human. Traditionally, there were 3 classes of cnidarians recognized. Now, nearly all biologists accept 5. So we have Anthozoa, the sea anemones, the corals, and sea fans. Hydrozoa, the hydroids called the hydra and the Portuguese man o' war, and then Scyphozoa, the jellyfish. So now, nearly all biologists accept some scyphozoans are sufficiently distinct in life cycle and morphology that they constitute their own class. Cubozoa, the box jellies, some of which have a sting sufficiently toxic to kill humans. Less widely accepted is the separation of some other scyphozoans into their own class. star rozoa, star jellyfishes. The bilateria, characterized by a key transition in the animal body plan, bilateral symmetry, which allowed animals to achieve high levels of specialization within parts of the bodies, such as the concentration of sensory structures at the anterior end. The bilateria is divided into two clades. One comprises the protostomes and the deuterostomes, which are discussed in chapters 33 and 34, which we'll get to next in this course. Here we briefly mention the other clade, the acyl flatworms. As its name implies, an acyl flatworm lacks a coelom. And there you have it. So the acyl phylogeny, the acyl flatworms, were once considered basal members of the phylum Padiominthes. This has been your instructor, Scholar Huff.